we're going to look at photosynthesis in two distinct stages. One is going to be sort of an energy, not energy creation, but taking that sunlight, turning it into usable energy. Then we're going to take that usable immediate energy and build molecules out of it. That building molecules part is going to be the light independent stage. So first we're going to look at this light dependent stage. Can't happen without light. This is going to take place in an electron transport chain, much like in cellular respiration. So if you've seen that before, particularly recently, that'll be very helpful to you because it's the same type of process. So we are in the thylakoid membrane right now, and there's two photosystems. Confusingly, these two photosystems, which are like a combination of proteins and chlorophylls, very complex, hundreds of molecules. Photosystem 2 is the first stage of the electron transport chain, but photosystem 1 was discovered first, so they're named in a confusing way, where it goes from 2 and then to 1, but that's just how they were named, unfortunately. So here's what it is, a more complex diagram of what it looks like. We are in the thylakoid membrane, so the membrane surrounding one of those pancakes. Out here on the top is the outside, the stroma, and then down here at the bottom is the inside of one of those pancakes, called either the thylakoid space or the lumen. So this will be important because we have a small contained region within this organelle. So here we go. We're going to start at photosystem 2. A photon, a packet of light energy, is going to strike this photosystem. It's going to be received by photosystem 2, and this will only receive particular wavelengths of light. When that happens, there's a whole process that happens. It's briefly described in your book, but it's basically exciting the molecules inside of this photosystem, and they're going to transfer that sort of excited energy from one to the other in a way that amplifies it. That amplified energy is then going to be transferred to a chlorophyll A molecule. All of our photosynthesizing organisms have had chlorophyll A because this is chlorophyll A's job. There are special chlorophyll A molecules that, when excited, will shoot off an electron, and that is going to get the electron transport chain moving. But a chlorophyll molecule that continuously loses electrons will cease to be a chlorophyll molecule. So we have to have a way to replace that electron. This is water's whole job. This is why plants need water, is to replace that electron on the chlorophyll. So what's going to happen is at photosystem 2, we are going to take molecules of water and break them apart. That breaking apart is going to release a low energy electron that will then go back onto that chlorophyll molecule, and the high energy excited electron from the chlorophyll molecule is going to go through the electron transport chain. So those are huge, big events in photosynthesis. This receiving of a photon, the excitation of a chlorophyll molecule to bump off an electron, and the replacement of that electron by splitting water. The other big thing that happens here that you'll notice is when we split that water molecule, we have made some hydrogen ions and some oxygen. That oxygen can be used about 10% within the plant to do cellular respiration. The other 90% is going to be lost just as a waste product out into the atmosphere. Why do these hydrogen ions matter? What do you know about hydrogen ions? Thinking about this week's lab and pH. pH is a measure of concentration of hydrogen ions. So as we build up hydrogen ions in this tiny little space, we are lowering the pH, confusingly, and making a more acidic environment. The other thing to know chemically is that positive charges or negative charges, the same charge is going to be repulsive, right? When you put two sides of a magnet that repel each other, that you can't force together, they have the same charge. If you were to flip one of those magnets over so that the opposite charge, it'll be attracted to it, right? So that's another thing that's happening here. we are piling up hydrogen ions that all have positive charges in the same space. So it's becoming more acidic, and those hydrogen ions are just jiggling around everywhere, repelling each other, wanting to get out of this space. A lot is happening. So let's follow that electron around. Oh, here's my little summary page. So we have our incoming photon absorbed, exciting the chlorophyll molecule. Chlorophyll molecule shoots off its electron. That goes into the electron transport chain. Water gets broken apart. This is oftentimes called hydrolysis. Hydro for water, lysis for breaking apart. So water is broken apart, and we get oxygen produced as a byproduct just in this very first part of photosynthesis. And we're also starting to build up these hydrogen ions. Well, I just talked about this all on that one page. Okay, so now the chlorophyll molecule has gotten its new electron from water, and it's ready to receive another photon. This whole process takes like a millionth of a second, some incredibly short amount of time. But a chlorophyll molecule can get exhausted, so a lot of times plants will have a way to rotate their chloroplasts around to kind of maximize the amount of sunlight they can receive. If I just said something untrue, I hope I didn't. I'm pretty sure that's true. So oxygen is just a waste product of photosynthesis, and we are building up those hydrogen ions in the thylakoid space. So now we get to look at the rest of our electron transport chain, which is what it says under here. We see our electron being passed from these different protein complexes across the membrane. These different protein complexes are, each has its own job. Two of these are proton pumps. So the electron here serves as a bit of a power source. Think about electricity as this transfer of electrons. So when you transfer electrons through these different protein complexes, you're basically plugging them in. You're giving them energy to do the job that they are designed, set up to do, right? So each of these protein complexes is going to take protons, these H+ molecules, hydrogen ions, and pump them into the thylakoid space. These protons, H+ molecules, ions, atoms, they're not molecules, can't move across on their own, even though they are basically as tiny as you can get. They can't move through this membrane because they have a positive charge. So charged particles can't move through membranes on their own. They have to be brought over. The other thing that's going on is there's already a lot of hydrogen ions in here. So whenever you're pushing something up its concentration gradient, putting it to where more of those things are, that takes energy. So it's like shoving them across the membrane, even though there's already a bunch of them over there. So that requires energy to do. So we're getting that energy originally from this photon, but then by the electron that gets transported across. So the electron is making this series of jumps and kind of losing a little bit of energy with every jump. So after we get to this point, photosystem 1, we have this pretty low energy electron. Okay, so hydrogen ions are pumped across the membrane into the thylakoid space, a relatively small area, and that is up or against their concentration gradient. So normally things flow from high concentration to low concentration. We are shoving them back the other direction, taking energy to do so. And the hydrogen ions hate it. They're too positive, they don't like it, ugh, they hate it. They really want to get out of there. Like so much so that it would be a great release of energy to do so. So by doing this, we're creating something like a battery, where we are taking a chemical concentration gradient, or we're building a chemical concentration gradient, putting these hydrogen ions into the inside of the thylakoid membrane, making it relatively positive, to this relatively negative space out here. So we're creating a battery across this membrane by having this concentration gradient and making it stronger. That's the whole point of the electron transport chain. So we have this low energy electron that is going to be transferred to photosystem 1. Here we are at photosystem 1, which is going to receive another photon. So that photon is going to re-energize that electron, and it's going to be transported through a few different protein complexes. The one that you would want to know the name of is this last one. And all of these are named in the textbook if you are curious about that. So, right here, we have this protein complex called NADP plus reductase. It's a pretty long name, but it tells you exactly what it does. So NADP plus is a compound that can, or a molecule that can accept electrons and carry them to another place. Now again, think about electrons as a form of energy. So NADP plus can pick up 2 electrons because it has a positive charge it can balance. Boom, that's 1 electron. And then it's also going to swoop up this hydrogen ion, which will also need to be balanced by another electron. So electrons are negatively charged. We can pick up 2 and we can make NADPH. Because we put electrons, which are negatively charged, onto this compound, NADP+, we have reduced it. We have reduced it by adding negative charges. So this enzyme here is called NADP+ reductase because it reduces NADP+ to NADPH. NADPH is now an energy carrying molecule because it's carrying these high energy electrons, and it's going to transport them to our second stage of photosynthesis. So the energy to power the second stage is partially being carried here. We're going to have a second high energy carrying molecule that we're going to form. So here we've taken this electromagnetic energy, we've excited some stuff, moved it around, formed chemical energy, and that chemical energy is being stored here in NADPH. We've also, through this process, formed an electrochemical gradient, and that is stored potential energy across this membrane. So far we've taken electromagnetic energy, turned it into chemical energy and potential energy by storing it across this membrane. The last thing that's going to happen in this electron transport chain, other important thing to note here, we are taking hydrogen ions out of solution in the stroma. So we are putting them in a high concentration inside the thylakoid space and reducing the amount of them in the stroma. So again, that's just strengthening this concentration gradient, making extremely low concentration of hydrogen ions in the stroma and a higher concentration of hydrogen ions in the thylakoid space, both by breaking open water and making those hydrogen ions, as well as pumping that those hydrogen ions across with the electron transport chain. Okay, the big thing that happens here is ATP synthase. Again, it says exactly what it does. This big pear-shaped complex here, this enzyme, is going to take ADP and a phosphate group and turn it into ATP. This is a high energy carrying molecule, which you know from cell bio, is that ADP has 2 phosphate groups, diphosphate, and each of those phosphate groups has negative charges. So they are already repelling each other, and it's not a happy molecule. To shove another negatively charged phosphate group on there is just like the least energy state that this ATP wants to be in. It would love to get rid of that 3rd phosphate group, so you really got to shove it on there. It takes a lot of energy to do so. So why do that? Because now you've created this molecule that wants to donate energy as soon as possible. So often when you're turning things on in a cell, it's called phosphorylation, and that's ATP coming in and dropping off a phosphate group, releasing that energy, powering that process. So this whole process creates high energy molecules called ATP. How does it do that? Well, think of it like a water wheel. And we have this high concentration of hydrogen ions here. We're going to let them flow through our water wheel or our turbine, and that is going to cause it to spin. This flow of protons are going to flow through, spin this ATP synthase, and that's going to give it kinetic energy, movement, and that movement is then going to be transferred into chemical energy as we slam that phosphate group onto ADP to make it ATP. So, summary of this process, we've used electromagnetic energy in the form of photons to then get some excited molecules and chemical energy. We've created a concentration gradient with stored potential energy, as well as some NADPH, which has stored chemical energy, which is also a type of potential energy. So then we're going to take our concentration gradient and spin an enzyme around with kinetic energy, and then take that kinetic energy that we made and whoosh, make some more chemical energy. So the whole end result here is that we have some chemical energy that we're then going to transfer to our second stage of photosynthesis. So this is a summary table that you can make. We'll fill out the second part of it after the second stage. But if you want to pause on this screen and write down where these events take place, what are our products, and what are the important events.